Welcome everyone to another episode of the Exocrine Gland Biology Podcast. Today we will be discussing a fascinating and relatively new topic in salivary gland biology, the Tuft cells. To teach us about this exciting area of research, we are interviewing Dr. Harim dos Santos, whose work has helped establish the presence and functional significance of Tuft cells in the salivary glands. Before we begin today's discussion, I would like to briefly introduce our guest. Dr. Harim dos Santos is an assistant professor in the Department of Oral Biology at the University at Buffalo, State University of New York. Originally trained as a dentist, he earned his DDS degree in Brazil and later completed a PhD in stomatopathology at the University of Campinas. Following his doctorate training, Dr. Dos Santos completed postdoctoral fellowships at the University of Utah and the University of Missouri, where his research focused on salivary gland biology, tissue regeneration, inflammation, and the mechanisms underlying salivary gland dysfunction. Dr. Dos Santos is particularly recognized for his pioneering studies on Tuft cells in salivary glands, including the first demonstration that these specialized epithelial cells are present across multiple species and subsequent work revealing their functional importance in the gland's homeostasis and disease. Harim, thank you for joining us today. Before we dive into the science, could you tell us a little bit about your scientific journey? What drew you to do salivary gland research and what you are currently working on now, please?
Morning. Thank you very much for the invitation and for this chance of talking a little bit about Tuft cells. They are pretty interesting. And about my scientific journey, I became a dentist in Brazil. Then I enrolled in a master and a PhD program in stomatopathology. And this is basically otopathology, oral medicine. And I had the chance during this time to be part of an international internship in Mexico and in Portugal. And after that, I participated in a residency in oncology for a year or so. And by that time, I received an invitation to be an instructor at a university in Brazil, at dental school. So I took the opportunity. So I had the chance of connecting with students. And by when I was 28, by that time, I was like, okay, I think I also I miss being a researcher because I was away from research for 2 years. And that's when I started applying for postdoctoral positions in USA. And I started working with Dr. Baker, first at the University of Utah and later at the University of Missouri. During my master's and PhD, I worked with salivary gland cancers. And during my postdoctoral fellowships, I had the chance of working with salivary gland regeneration and inflammation. And by that time, that was when I got a grant for the drug from the Drogen Foundations and from the NIDCR, a K99, and I had the chance actually of building my own lab after that here as assistant professor at the University at Buffalo.
That's so interesting. I didn't know this story. And I like how we shape our career based on opportunities. But then you said I missed the research, so I decided to go for it too. And now I have a question. Do you miss the clinic?
Since my undergrad, what drew me most was the understanding why diseases happen. So, although I felt like attracted by the clinic, for sure, what attracted me the most was a microscope. So I think when I was like in my dentistry course, 2.5 years already studying dental school in the dental school, I thought, you know what? I think I love autopathology. That's what I want. And that's why, like, back in 2009, 2010, I was like, that's what I want to make for as a way of living. And so I miss a bit the clinic, but definitely. I'm really satisfied living what I'm living. Sometimes I think that I'm living the American dream. I'm doing what I love. I'm in a place that people support me so much, and I'm paid to do this, to do what I like. So it's really like a unique experience that I feel that I'm really fortunate and grateful for being experienced in this life journey, right?
Yeah, that's amazing. So let's talk about the oral pathology now, the Tuft cells. So, for who doesn't know or are not familiar with the cells yet, what exactly are the Tuft cells? What are they known for? Where are they in the body? And what first made you suspect they could be in the salivary glands too?
Okay, so Tuft cells were discovered 60 years ago. I think it was first in mouse trachea and later I think was human stomach. And believe it or not, their function remained unknown until 2016, until three independent groups showed that these cells have immunomodulatory role in the intestine. And after that, different groups showed that these cells also have different kind of functions in different tissues. In the salivary glands, these cells were pretty well described in rat salivary glands in the 80s, 90s, and early 2000s. But when I decided to go for this as like an idea, I've noticed that the last publication of cells in salivary gland was like 10 or 15 years before my actual publication identifying them in mouse, pig, and human salivary glands. And this was in 2021. And how did I discover them? Well, it was actually pretty incidental. I was looking at some transmission electron microscopy images from the lab where I was during my postdoc. And I was just randomly checking the images. They were like 10 years old. And at one moment, I saw one cell that I saw present in a duct and had like well-developed microvilli. And I thought, okay, I don't recall ever reading about. I'm not sure if this image is actually I don't know, some kind of artifact. And I actually went to Google or PubMed, and I think I typed it like cell big microvilli duct. That's when I found about the tuft cells, and then it was like tuft cells, salivary glands, or they were also called brush cells. There are different names, and I've noticed that there wasn't like a specific publication in actually human salivary glands, for example, say, oh, where they are also not on pigs or mouse. That's when, after discussing with my mentor during the postdoc, I said, why don't we just do like a paper showing that they are there? And I think the mentor gave me this opportunity to actually go for it. That's when we found there. We use a transmission electron microscopy. We use a confocal. Nowadays, I have some data with a spatial transcriptome showing that the cells are for sure there, as expected, by the way. And now we are trying to understand better what is their role. Now that we established that they are there in different species, we want to answer the question, what are they doing? Because in different organs, as I mentioned, they have like immunomodulatory roles, or actually in intestine, they are also shown to be related to regeneration. In pancreas, they are being shown to be related to carcinogenesis. And the question is like, what are they doing in the salivary glands? And that's what I'm trying to address here at UB.
Very nice. Do you know the word serendipity? When you find something by luck, you're not looking for it, but you find this. Like the discovery of penicillin, it happened like this. And it's the same for you. I like that.
Oh, yeah, I'm not comparing this to penicillin that
I
you
know.
have, but for sure was by chance. I was just like, I don't know, listening to some music and seeing the image, trying to see if I could find anything that could be, you know, worth seeing,
Yeah.
or if I could find a beautiful picture. And yeah, so that small like moment doing this now helped me to actually change my scientific path, and
Yeah. That's very nice. And I saw that you have two main papers about this now, one from 2021-22 and one very recent from 2026. And the first one, you demonstrate the presence of the cells, and you were the first author of it. And then in the later one, you demonstrate more about the function of the cells, and you are the last author. So could you talk a little bit about this scientific journey when you start being a trainee and then you become a PI?
Oh yes, I think when I was a postdoc, I mostly my role was to execute. And now, as a PI, my role is to design, to architect the experiments, to build a team, to make the questions, try to address the questions. I think is another level of responsibility when I compare with my postdoc. And during my postdoc, I answer the questions. Are they there? The answer is yes, they are there. Now, during my independent phase, I'm trying to answer the question, what do they do there in the salivary gland? So I use a mouse model lacking POU2F3 that it is a master transcription factor for tuft cell differentiation. This is for sure one of the most widely used mouse models to study tuft cell functions in different organs. So I compare salivary gland architecture, transcriptome, function between wild type and knockout mouse, male and female. And what the group observed was that the morphology of the gland is different. In the knockout mice, there is a degeneration of ductal and acinar structures. There is an alteration in the transcriptome and in the saliva quantity and composition. What is pretty interesting is that this alteration in the transcriptome is sex bias and showing that in the female, most of the alterations due to tuft cell loss is related to transcriptional changes. While in the males, what you mostly see is like an inflammatory upregulation in the mice lacking tuft cells. Interesting, the saliva of both sexes lacking tuft cells, the saliva quantity was increased when compared to the wild type mice. This was unexpected, but what it does indicate is that these cells, at some level, may be involved with the regulation of saliva secretion. And this would not be a surprise, actually, because in different organs, like the intestine, tuft cells are directly related with epithelial secretion. Believe it or not, they produce and secrete acetylcholine. And as we know, like the people from the audience of this podcast know, acetylcholine is essential for saliva secretion. On the way to understand better how actually these cells in the salivary gland may have any role in regulating the secretion, saliva secretion in this organ.
From these findings, what was the most surprising for you?
I think I think I have two surprising. Like, was the sex bias was like really striking.
Mhm.
Um, of course, I know that there is like sexual dimorphism in the salivary glands, but I didn't expect to be to that extent on the role or on the impact of tuft cell loss. And also the higher saliva secretion was also a surprise. But yeah, that's science, right? It's
Oh
not
yeah.
always as we expect. Um, but what was good, for example, what is a good surprise? The third one was when I saw that when I looked the transcriptome results and the mass spectrum results of comparing wild type to knockout, I saw all the wild type grouping in a heat map, all the knockout grouping separated from the wild type mice in the heat maps of, as I said, Bucharest and mass spec. I was like, okay, this looked like really pretty sharp. You see a clear pattern there.
Mhm.
So that's why we are trying to research more on this.
Nice. Do you have any limitation of the model that you think it's important to acknowledge in your studies?
Oh yes, so this is a global knockout. It means that mice lacking PO2F3 lack tuft cells all over the body. So one limitation is, are these difference in the transcriptome morphology or saliva quantity and composition primary or secondary to this? So what I'm trying to do now, I got a mouse model that will allow me to directly ablate the cells only in the salivary glands.
Oh, nice.
So it was a donated mouse model. So now we're gonna get actually the answer to this at only the salivary gland level.
Are you using AAVs for that?
Correct. I'm in this exact point trying to choose what is the best AV to induce the local ablation. We got PO2F3 flux mice from Monell Institute. So it was like, I'm really grateful for this. It saved a lot of time and money.
Hmm.
And now, as I said, we are generating the AAV so that we can start injecting and see if we can ablate locally to remove this limitation of the knockout mouse model.
Nice. Something that I always say in this podcast, it's about the collaborations, how it makes a difference. So you're just telling me that you got this animals from another group and how that can help you save time and money. And I think that's amazing.
Yes, collaborations are the key. I think not only in our profession, in any profession, we need to know people so that we can help and
Yeah.
also be helped.
Work together, right?
Mhm.
In the same study that we're talking about, the sex bias, you also used human samples and you found some associations between tuft cells abundance and disease-related features. Can you talk a little bit about this?
Yes. What we did in this experiment, we compared Tuft cell abundance in no Sjögren Zika patients and Sjögren's patients. And what we saw was that the number of Tuft cells was correlated, for example, with inflammation, with saliva flow rate, with focus score. And what is really interesting is that in the organs where Tuft cells are found, there is a difference in the abundance of the cells when they modulate the inflammatory environment. That's where the idea came from. It was like, okay, in different organs, we know that there is a difference in the number of Tuft cells during inflammation in mouse models and also in humans. And my question was like, okay, maybe in Sjögren's patients, well, what can we see? What is very interesting, Juliana, is that there are some patients in both groups that don't have Tuft cells. Some patients in both groups have a low number of Tuft cells. Some patients in both groups have a peak on the number of Tuft cells. And my question is like, why? Why do we have some patients without, some patients with a lot? So that's why I'm actually trying to address now. I'm getting samples from all these groups and try to compare there at the transcriptome and proteomic level and see what are the difference. Does the epithelial cells or the inflammatory infiltrate change in patients with more Tuft cells? Or is it the same? In the mice, for example, we saw that the loss of Tuft cells led to alteration in saliva flow rates, in saliva composition. My question is in patients that we don't see Tuft cells in the salivary glands, do we also have an alteration in saliva composition? That's what we are trying to address with, again, with collaborations. They're like being very helpful in providing samples for the analysis.
And if we go back to your first paper, because now you're saying that you found something in mouse and then something in humans, and you want to understand it better. And in your first paper, you addressed the tuft cells in three different species, right? Is there a reason why you decided to do the three different ones?
Oh, the reason is pretty simple. The idea was to check if a rare cell type, by the way, was a conserved component in salivary gland biology. And we know, for example, if we have just the cells in the mouse, but we don't have in human, the translational relevance it basically is really hard to explain to even in a paper or to submit a proposal why I'm researching something in salivary gland biology with the ultimate goal of helping humans that is not in humans. So I think we wanted to address because mice would allow us to do most of the subsequent experiments. Pigs, we use it because we know that pig salivary gland share some similarities with humans. And of course, humans are the ultimate goal to determine the translational relevance. And currently, we are fortunate enough to be obtaining human samples, minor and major salivary glands from different collaborators here at Buffalo and also outside the country and in different institutions in USA. So it's been a journey, but pretty good so far.
And as you said, those cells are rare. So did you have any biggest technical challenges in proving that the cells were present in the salivary glands? And looking back now that you have all this experience, would you do something different in the beginning?
Yeah, I think what was the most difficult piece of data to obtain was the transmission electron microscopy images, especially the ones from the last paper. Getting the beautiful microvilli, the apical tubovesicular system, the lateral projections, very clear. This was challenging because these are really rare cells, less than 1% of epithelial cells in baseline conditions. So it took me a long time to actually find good, really good image. I could find, for example, some cells where you saw part of the tuft cell or part of the morphology, but previous papers actually in transmission electron microscopy focusing on tuft cells say that their position in the duct is diagonal. So as it is diagonal, when you cut a section, it's going to be hard to find the whole morphology of the cell. That's why it took me a while to find in mice, in male and female, like really, really, really sharp images
Mhm.
that for sure show tuft cells in accordance uh, with tuft cell morphology ultrastructure in different organs. What I would have done differently, I think I would have focused more on maybe in a single cell analysis right on the first months of having this starting working of this. However, that's what we are working now, and hopefully we get like really good data out of it. But that's one of the things that I think would have clarified some other questions like faster.
For the transcriptomics, you are doing the single cell, right?
That's where we are working now. We are working with the single cell to try to isolate this cell population. And also, we are trying to establish a mouse model where we have an increased number of tuft cells to be easier to isolate them.
Oh,
In other
wow.
organs, for example, in the intestine, what you have like um, an infection with some microorganism, and tuft cells that represent 1% of the cell population there have an increase of 15-fold in their numbers. Then it's really it's easier to isolate. Also, in pancreas, they there you can do a drug treatment to increase the number of tuft cells and allow you to isolate the cells. That's what actually a lot of groups do. And actually, I think maybe this week we got it.
Okay.
As I'm really excited. Yesterday, we're still confirming results, but I really think we finally got a mouse model on which we get a, increasing number of tuft cells and we allow easier isolation to study better, the transcript, the single cell features. And yeah, fingers crossed it.
Yes.
But after a few years, I think finally we got something very easy directly in the salivary gland, you
Yeah.
know. So yeah.
So I hope you get this model working and you can get it easier because I know it makes a difference.
Hmm.
So let me see if I understood the path. Does two works on Tuft Cells are part of your K99?
No. The first one I published the one I think was 2021,
Mm-hmm.
and then I got the K99 I think 2 years after. Yeah, 2 years after I submitted the proposal. This was while I was in Missouri. Then I resubmitted the proposal, and that's when we got the K99. But for sure, it served as basis. It's like in the innovation section of the proposal, I put that this is innovated because the last publication on this in salivary gland was like 10, focusing on this was 10 years ago. There you have a knowledge gap there, and you don't know the function. They are there for a reason. But yeah, first that paper, then K99, then I came here to UB. Um, that's when I activated the R00, and now we are working towards more determining the function. The role is specifically in inflammation. So that's why we use different models of inflammation, like injecting with LPS or Sjögren's mouse model to address these questions. Actually, there is a mouse model called the non-obese diabetic, the NOD, and what we did, we removed the tough cells from this mouse model, and we are still collecting the samples, and we are trying to see if there is a change in disease progression in the mice lacking, in the NOD mice lacking tuft cells. So this is all ongoing. It takes a while because I collect male, female, and 4 different time points. We collect serum, salivary glands. So it takes a long while. It's years of work and then like a while also to analyze.
I'm excited to see these papers coming out soon.
Hopefully.
With this, how do you see the field evolving over the next 5 to 10 years, considering that everything takes a lot of time?
Yeah, I think initially is more, the current studies are more descriptive. Now we are going more mechanistic, understanding why, how. So I think that's the future, is understanding white of cells, like what they are doing, what is the origin, you know? For example, in different organs, I think it's trachea and lung, Tuft cells and ionocyte transdifferentiate.
Hmm.
So this is very interesting. But however, in the intestine, as far as I know, there is no ionocyte. So it means that the cells there don't need an ionocyte to differentiate. So my question, like, what does it happen in the salivary glands? Where do these cells come from? So this is for sure years of research if you go this way. Or you can understand better the role of the cells in different injury models. Like I'm focusing on inflammation. But also the question, do the cells have a role in regeneration after radiation?
Mhm.
So I think there is a lot, a lot. I just started to get some pieces of the puzzle together, but there is a lot to understand why, how, when,
Mhm.
and I think that's it. I think it will go the evolution of this field in Tuft Cells and salivary gland biology is from descriptive to mechanistic, and hopefully with some clinical applicability to the to humans. Because as we discussed it a few minutes ago, we saw that there is a difference of the number of these cells in patients with Sjögren's and non-Sjögren's Zika patients. So like can these cells be used as a biomarker of the disease? What they are doing there? You know, currently the findings that we have show that the cells possibly have an association with Sjögren's, but association with is not the same as causation. So we don't know if they are there as a consequence or as a cause of the disease. So that's why what we are trying to address now.
Nice. And based on all this, could you guess what is the primarily function, if they're sensor cells, regulator, immunomodulators, or something else in the salivary glands?
Oh, our preliminary data are indicating that they are pretty much chemosensory,
Okay.
similar to tuft cells in different organs, you know. But like now, what we are trying to address better is like what are they sensing specifically? You know, what are they sensing? Are they sensing bacteria? Are they sensing difference in pH? Are they sensing inflammatory mediators? At this moment, I don't know. That's why I'm using this grant to funding these studies to determine what are they sensing and what happens when they sense something. And yeah, I hope to have maybe a few years from now if we have another chat, I can come with some more definitive
And
answers
I'll
or more
invite you
questions.
again in the future so you can bring me all the answers that we cannot answer right now. And for the students and postdocs and early career research listening today, and they want to enter the field, do you have any advice that you would like to share?
Oh yes, never give up. You need to be sure that you're going to receive more no's than yeses. It is part of the game. You know, when I wanted to become a postdoc in USA, I sent a bunch of mails to people all over the world. I sent a total of 124 emails, and just one person gave me the chance to have an interview. So, and I got the position,
Wow. Yeah.
and here I am, you know. Uh, but you need to be very determined, you know. It's like, and it's understand, it's gonna be more no's period, you know, and don't take it personal. Just keep going, be determined, know your purpose, know your goals, have discipline, you know. Sometimes, yes, it is a lot. Sometimes it is a lot. Sometimes it can be overwhelming. Just like try to achieve some life work balance is also important, is essential, you know. There are some days that I'm just like, you know what, I'm not going to work this evening. I'm gonna just have a walk, have a beer, and live. But yeah, I think the number one thing that I would say that I think it's what determined my journey, but doesn't necessarily apply to everyone. I feel that I'm pretty determined. I don't take no as the final thing in my life. I don't get offended by it. I just see it as a step for something else.
And before we finish, is there anything you are currently work on that you are excited about, or something that you'd like to share, a conference that is coming up, something in that sense?
Oh, currently I'm working in some spatial transcriptomics data sets, comparing like different sexes. And I think this is something really, really exciting that I'm working. I'm also collaborating with different groups. They are being essential, and I'm really grateful for their essential help on this. And I think it's something that is giving me a lot of headaches, but also is keeping me, motivated and try to put together and trying to get something nice that can be useful for other people. And yeah, I think this would be the thing number one that the lab now is currently working, and hopefully in a few months from now we can put out there for everyone to read and appreciate or criticize and open to all.
I'm looking forward to seeing this out. Well, with this, I want to thank you so much for joining us today and sharing your research, your expertise. It has been a pleasure learning more about the Tuft Cells with you and the exciting discoveries that are coming up soon. I hope you all the luck, and I have my fingers crossed that everything you have been working on is going to work well and give you the answers you need.
Thank you very much. Again, I appreciate the invitation, and yeah, I wish you all the best as well. Hope to see you soon in some conference, and we go from there. Thank you.
Thank you. Bye bye.
Bye bye.
To our listeners, thank you for joining us for another episode of the podcast. We hope to see you soon. Bye bye.